Finding new cancer biomarkers behind disease progression in IgM, MGAS, or WM is sometimes challenging. We now know that the MYD88 somatic mutation is present even in lymphoid progenitors, as it has been recently reported. But this mutation partially explains the expansion of this lymphoplasmacytic clone that can cause further disease progression. And we are still working on finding new cancer biomarkers behind disease progression, which we think is very important and crucial. But we also acknowledge that it can be challenging because of the long times of latency that we can see in the clinic in these conditions. So in this project, we hypothesized that understanding how a B-cell clone expands in a patient can give another mechanistic view and help us to find novel biomarkers. So to do this, we performed single-cell RNA sequencing paired with the PCR data from each single B-cell. And in some other set of patients, we performed single-cell ATAC sequencing. We put the attention on this assay, the single-cell ATAC assay, that consists of looking the chromatin accessibility landscape, the chromatin states. So after filtering the whole set of cells that pass the good quality control filters that we analyze in our data, we obtain approximately 135,000 cells, and we looked that the cell of origin in most cases was associated with a memory B-cell phenotype. We then analyzed the BCR sequences of each single B-cell, and we were able to delineate 3 important clones. And actually the most important was the hyperexpanded or tumor clone. The other 2 smaller clones were actually either emerging clones or probably non or unspecific clones that we can see sometimes after exposure to acute or chronic inflammation. So this hyperexpanded or tumor clone displayed memory B-cell phenotype gene expression signatures, and regarding the chromatin accessibility of this hyperexpanded tumor clone, we observed that there were open chromatin regions specific to this clone. And in fact, we found that the ETS1 and FOXP1 transcription factors were highly enriched. These transcription factors have been previously reported in ABC-DLBCL, suggesting a high dependency on the downstream signaling of the MYD88 somatic mutation and the BCR signaling. However, the most important transcription factors were the POU family of transcription factors. This consisted of POU2F2 and POU2AF1, that is a coactivator protein. We think that they behave as scaffolds, and they build like an epigenetic network consisting of multiple proteins that cause an increased accessibility in the chromatin of these hyperexpanded tumor clones. And they were very specific to the tumor clone in contrast to the other normal B cells. So I think the most important finding is this POU2AF1. Now the clinical translation of this transcription factor is that it's being reported nowadays that it can be a target by degrading the proteins that interact with this transcription factor in treating DLBCL, other mature lymphoid neoplasms, and even multiple myeloma. And we think that it can be also helpful in Waldenström's disease.